For the first time, we have established a modification of the genes coding for inotropic GABA-A and metabotropic glutamate receptors in subsets of neurons in the adult honeybee brain.
CRISPR-Cas9 gene editing can be used to modify one or multiple genes in adult bee brains to explore their roles in learning and memory under controlled laboratory conditions.
This method can be used to study the function of specific proteins in adult bees, as well as to test the specificity of antibodies against their corresponding proteins.
Demonstrating the procedure with Irina Sinakevitch will be Lev Kurtzman and Hyun Choi, former undergraduates from my laboratory.
To test the expression of RDL and mGlutR1 by immunocytochemical analysis after CRISPR-Cas9 injection, first, use an online CRISPR-Cas9 tool to design the guides using the guide DNA sequence of Apis mellifera RDL and mGlutR1. To prepare guide RNA complex formations for each guide RNA, label one test tube for each guide with the name of the guide RNA, and add 92 microliters of nucleotide-free buffer, 4 microliters of 100 micromolar fluorophore-labeled transactivating CRISPR RNA, and 4 microliters of the appropriate guide RNA solution to each tube. When all of the materials have been added, mix the solutions gently, and spin the tube contents in a benchtop centrifuge for 5 seconds to sediment the solution. Then, heat the solutions to 95 degrees Celsius.